So our next speaker is Jim Udelson, Chief of Cardiology at Tufts and Editor-in-Chief of Circulation Heart Failure, who is going to present incorporating Infarct Size into trial composite endpoints, implications for unloading trials.
Great. Thank you very much. It's really a pleasure to be here. So in the next few minutes, I'd like to address the issue of running clinical trials in this area, particularly as you move into these more non-shock, high-risk patients. And we'll first talk about the problem with doing trials like that, and then we'll talk about biomarkers as surrogates, and then how to incorporate markers into endpoints. So the problem is that in contemporary practice, many people do pretty well. And in the heart failure world, where I usually live, you can see that in the paradigm trial of valsartan and sacubitril, the 1-year risk of cardiovascular death or heart failure hospitalization was only 10 to 12%, and that's why you needed 8,000 patients to do this trial. And the same thing holds true in STEMI trials. These are just some data from a relatively recent trial, AIDA-STEMI, intracoronary versus intravenous abciximab. The primary endpoint, all-cause mortality, recurrent MI, or new heart failure. So even with that 3-point composite, took 2,000 patients, the event rate was only 7% at 90 days, and therefore the vast majority of patients in these trials do not contribute to the endpoint. So enormous amounts of money are being spent. The vast majority of patients do not contribute to the primary endpoint. Now, of course, if you study patients with large anterior myocardial infarction or certainly cardiogenic shock, that's different. But still, as you try and move a therapy like unloading into less severely sick patients, these event rate issues will really hold. So there's a lot of interest in trying to figure out how to get biomarkers of some sort as surrogates into these trials. And when I talk about biomarkers, I'll talk in general, and I mean either serum biomarkers such as troponins or natriuretic peptides, or imaging biomarkers such as Infarct Size, which is really what we'll talk about today, left ventricular volumes, or ejection fraction. But even though we'll talk about Infarct Size, the concepts really are across the board here. So what makes a marker like that a good surrogate? And these are just a summary of the classic papers of Prentice and Demetz and Tom Fleming here from quite some time ago, that the surrogate marker, number one, is in the causal path between the therapy and the outcome, and that all intervention effects pass through the marker in the causal path or are captured in some way by the marker. Now, certainly if we're talking about myocardial infarction and infarct size by cardiac MR or sestamibi imaging, for instance, as the marker, that's pretty plausible. Like many of the outcomes, not all, but many of the outcomes pass through infarct size, or infarct size influences cardiovascular death and certainly heart failure hospitalization. And that's very plausible, and there's lots of data to support that. But where virtually every marker gets hung up is what comes third on this list from these papers, is that a change in the marker, so a therapy-induced change in the marker, is reflected in a change in the outcome. And there's virtually no marker, no matter what we think about prognostically, that hits this standard. And that's what I'd like to talk about next. And it's really important to understand that biomarkers that are prognostic are not necessarily good surrogate markers regarding effects of therapy. Those of you who have been around for a while remember PVCs, post-MI, were very prognostic, but suppressing them in the CAST trial increased mortality. So that was a marker where the therapeutic effect on the marker went in the opposite direction in terms of the outcome effect. And more recently, of course, we all know that HDL is very prognostic in terms of incident coronary disease, but raising HDL, in this case with torcetrapib in the Illuminate trial, increased mortality. So a change in the marker, even though it's prognostic at one time point, does not always translate into a change in outcome in the same direction. So this last point is really the one where if you talk to the FDA about incorporating surrogates and markers, this is what they'll tell you. Bring me the broomstick of the Wicked Witch of the West. Show me that a change in the marker is associated with a change in the outcome. And there's not too much data to support that. So over there in the poster session, we presented some data that I think hits this point for Infarct Size. And this was a patient, you can see the details over there, and I'm happy to talk to you later. a patient-level meta-analysis of 10 STEMI trials that we put together with investigators in the US and Europe, over 2,600 patients. The infarct size was measured by core labs, by either sestamibi imaging or CMR. We had 1 year adjudicated outcomes from all the trials, and essentially the message was that if you look at the adjusted hazard ratio for the treatment effect on infarct size, it had a relation with the treatment effect on heart failure hospitalizations. For every 1% reduction in infarct size in the studies, there was a 16% reduction in heart failure hospitalization, but no effect on all-cause mortality. So at some point soon, we'll be meeting with the FDA to ask them what they think about this, and can we incorporate infarct size into trial outcomes. Now, let's say they say yes. How would you do that? So in the last few minutes, that's what I'd like to focus on. How do you incorporate markers into endpoints? And so I think the best way to do this is by the use of these hierarchical composites. And the two examples that are most widely used and have the most potential, I think, are the Finkelstein-Schonfeld method, which Diane Finkelstein and David Schonfeld wrote about in Statistics in Medicine many years ago. They're statisticians in Boston. And the win ratio, which Pocock wrote about more recently in the European Heart Journal. Now, these have been used, as I'll show you, in the TAVR partner trials, and Tim Henry used the win ratio in his ixmilocel study published in Lancet recently. So what are these? So these are hierarchical comparisons of events or the timing of events in pairs of patients from the groups in the trials. And I'll show you pictorially what that means in a minute. The hierarchy accounts for clinical priority. Death is more important than heart failure hospitalization, and both of those are more important than a marker that you might put in the third spot. It allows the potential incorporation of longitudinal measures. So in heart failure trials, the change in 6-minute walk time, for instance, or biomarkers. But most importantly, the use of these types of approaches enables every single patient in a trial to contribute to the endpoint instead of only 10 or 15% of the patients. So let me show you my understanding of this as a non-statistician. So this is the Finkelstein-Schonfeld method. So let's say you're doing a STEMI unloading trial, randomized controlled trial, and your group A is 100 patients who get randomized to the unloading therapy, and your group B is the control. And let's say you have 100 patients in each group. So you're comparing pairs of patients. So you take, let's say, patient number 1 from group A and patient number 22 from group B. And you start with the first level of the hierarchy. Did either of those patients die? So if the patient in B died, group A wins that one comparison. And in fact, if both patients died, but patient A died later at 12 months instead of 8 months, A actually wins that comparison. That's how to wrap in timing here as well. But as I showed you, and as you well know, in heart failure trials or acute MI trials, most patients don't die. So chances are that neither patient will die, so you move on to the second part of the comparison, which is, let's say, heart failure hospitalization in the case of unloading trials. If B was hospitalized, A was not, A wins that comparison. But again, in general, particularly all comers in a STEMI trial, most of the time, neither of those things will have happened, 80 to 90% of the time. That's when, in theory, you move on to your marker that has a plausible relationship with outcomes. So if group A, the infarct size by cardiac MR was 5%, and in group B it was 12%, A wins that comparison. And so you have 100 patients in each group. You compare each patient in A to each patient in B. So you have 10,000 comparisons. And then you go ahead and statistically compare them, and you come out with a winner and a rate ratio and a p-value. And this is not foreign to the FDA, actually, and to the device group. This was actually the approach that was used in the TAVR Partner B trial, as some of you may know, that had a co-primary endpoint of all-cause mortality, but the other co-primary was the hierarchical composite of death and recurrent hospitalization analyzed by the Finkelstein-Schonfeld method. It was not a classic time-to-event method. So the concept is not foreign to regulators. The idea of putting a marker in the third spot, we are discussing it with FDA, and I think as we get into these types of trials, this would be a very important thing to establish with regulators in the United States, in Europe, and elsewhere. So basically, the message I wanted to leave you with was that incorporating validated markers into hierarchical composites may allow reasonable sample sizes, particularly for expensive devices, for trials of new approaches to ST elevation MI and high-risk patients, such as unloading. Thank you very much. Yikes, I was so spooked by the 10 minutes that I went way too fast. Okay.
But we were running late, so it was excellent.
Perfect.
So it was perfect. Is there one question from the audience? Regarding infarct size, there's a huge discussion what method you should use, whether you should use nuclear imaging or MRI for assessing infarct size and salvage area. Do you have any comments to that?
Well, my other hat is a nuclear cardiology hat, so I'll say cardiac MRI, actually. I mean, it really, it's obviously a more precise method, smaller standard deviations, which allow smaller sample sizes. On the other hand, when you start to do broader trials, the expertise to do it well, no matter what we might think at our centers, it becomes a little more challenging. So if you can get it done, that's by far the best method, I would say. If not, then there's certainly a tremendous literature about SPECT sestimibia imaging. Standard deviations are larger, the sample sizes will be larger, but the methods were established in the early 1990s and they've been used in many, many trials.
They're stable. I mean, there's much less inter-observer variability.
There's probably less with MR. I would say. It's more that it's just a less precise method, so the standard deviations are larger.
Thank you.
All right, thank you.